Hello everyone, I'm Charlotte Adams. I'm a PhD student at the University of Antwerp, and today I will be presenting how we reprocess SARS-CoV-2 human protein interaction data with the goal to gain new biological insights needed to combat a SARS-CoV-2 infection. As a response to the COVID-19 outbreak, Gordon et al. Published a SARS-CoV-2 protein interaction map. They performed affinity purification mass spectrometry on 27 SARS-CoV-2 proteins and identified 332 interactions with human proteins. Here you can see a couple of examples of SARS-CoV-2 proteins interacting with human proteins. In general, viruses have a relatively small genome and heavily rely on their interactions with host proteins for their survival and propagation. For example, for cell entry, interactions with host proteins are usually required. Also, for post-translational modifications like phosphorylation, SARS-CoV-2 needs to interact with host kinases as it does not encode any kinases. By studying these interactions, interesting drug targets can be revealed. For example, for the kinases RIPK1 and CK2, inhibitors exist that could be used against the SARS-CoV-2 virus. In the original study, proteins were identified with max_quant, and after protein interaction filtering, they achieved a protein interaction network. We reprocessed their data with open modification searching with Ansolo, and in addition to identifying proteins, we identified post-translational modifications. After a similar protein-protein interaction filtering step, we got a protein interaction network enriched with modifications. max_quant is a standard sequence database search engine that identifies unmodified peptides very efficiently. However, each variable modification that you want to study needs to be specified explicitly. The number of modifications that you can consider is limited because for each modification that you add, the search space increases drastically, which increases the computational load and decreases the sensitivity. Because only a limited number of modifications can be considered, often the most abundant modifications are chosen, which are artificial modifications induced during sample processing. With the open modification searching approach, both modified and unmodified peptides are identified. This increases the total number of identifications. Any number of modification, any type of modification is considered implicitly because the wide precursor mass window allows a modified peptide to match against an unmodified variant. This increases the total number of modifications and also the variety of modifications that are observed. So not only artificial, but also biologically relevant modifications are identified, revealing biological insights. Open modification searching has gained popularity in recent years because of novel algorithms that have made it possible to perform open modification searching efficiently and accurately on a large scale. Ansolo is a spectral library search engine that uses optimized approximate nearest neighbor indexing to speed up open modification searching by selecting only a limited number of the most relevant library spectra to compare to an unknown query spectrum. Ansolo performs open modification searching very fast and accurately and can sensitively match modified spectra to their unmodified counterparts. With the same false discovery rate of 1%, we identify more than twice the number of peptide spectrum matches in comparison to the original analysis. We reproduced about 80% of the peptide spectrum matches and 65% of the additionally identified peptide spectrum matches were modified. This indicates that the increase in peptide spectrum matches that we observe can be accredited to the ability to identify modified peptides. During affinity purification mass spectrometry, a protein of interest, also called a base, is expressed and purified. Each protein that is identified in the run can be considered as a prey interacting with the bait. However, affinity purification mass spectrometry data contains a majority of false positive interactions, a main source being non-specifically binding proteins. The protein interactions need to be filtered to distinguish the high confidence interacting proteins with the background. In most of the protein interaction filters, they start from the assumption that the background is constant across runs and that a interacting protein can be found by looking for proteins whose abundance is above their abundance across many runs of unrelated baits. We identified over 25,000 unique bait-prey pairs, of which only 375 remained after filtering with both missed scoring and Saint Express. Even though we use a similar protein interaction filtering approach as in the original analysis, we only reproduce about 49% of the original protein interactions. An explanation for this small overlap could be that after reprocessing the data, the observed background is different, causing a difference in which bait-prey pairs are able to pass the filters. With open modification searching, you can look at any type of modification, and we decide to take a closer look at phosphorylations. It is one of the most common and important post-translational modifications, essential for the regulation of cellular processes, and it plays an important role during SARS-CoV-2 infection. For example, a study found that the SARS-CoV-2 N protein needs to be phosphorylated in order to be able to interact with the 14-3-3 dimer. This is one of the highest expressed human proteins. We identified several phosphorylation sites on the N protein, but also on other SARS-CoV-2 proteins. Some of these sites were reported by proteomic studies, and most of the sites could be expected based on site prediction tools. We identified 2 novel phosphorylation sites on the SARS-CoV-2 protein Nsp12. Here you can see its interactions with human proteins. We reproduced 2 interactions with human kinases and additionally identify its interaction with a human kinase that was never reported before. After kinase-specific site prediction, we found that one of the phosphorylation sites, namely threonine 21, is predicted to be phosphorylated by TLK1. J54 is a new inhibitor of TLK1 and is currently being tested as a prostate cancer therapy. It could also be used to inhibit the phosphorylation of Nsp12, but the effect that this would have on a SARS-CoV-2 infection remains to be seen. To conclude, with advanced algorithmic approaches, we were able to identify more than double the number of spectra, and we identified 211 novel protein interactions. We revealed novel biological knowledge by providing a link between an interaction with a kinase and the observation of a phosphorylation on a viral protein. With this approach, we better understand viral infections and we reveal drug targets that can be investigated further. During this presentation, I only presented an example from our investigation of the phosphorylation of SARS-CoV-2 proteins. However, we also investigated the effect of ubiquitination and S-node translation to gain insights into how SARS-CoV-2 hijacks the cell. Thank you for your attention. Feel free to contact me and I look forward to your questions.